Good afternoon. So it is my privilege to introduce our last speaker. Professor Grzegorz Basak is professor in medicine, chair of Department of Hematology, Transplantation and Internal Medicine at Medical University of Warsaw, former chair of Transplant Complication Working Party of the European Society for Blood and Marrow Transplantation, and then current chair of Artificial Intelligence Subcommittee of that organization. His initial immunological background was experimental cellular immunotherapy in animal model. But currently, Professor Basak is leading the CAR-T program at his institution and developing clinical trial protocols in this field. He co-authored several papers on CAR-T cells, including European guidelines. Besides of being outstanding clinician, he's internationally recognized scientist and an author of almost 200 papers with really high H index and near 4,000 citations. So, Grzegorz, the floor is yours.
Thank you very much for a very kind invitation. It's an honor for me to be here at this research institution. It's really something for me because I'm also coming from the scientific ground, but currently I'm the physician. I'm doing some kind of physician studies. And my goal of my talk is just to show you, to my understanding, research as the clinical perspective, what we need, in fact, how we look at the problem of treating patients with CAR-T cells, which may be very important for those who develop those therapies, to maintain the communication and good collaboration, which is already very, very good. Okay, so of course, we doctors 10 years ago, probably just a few people in the Hematology Society in Poland believed that CAR-T cells will become our everyday treatments. And this treatment was neglected. This will never happen. Of course, it happened. We, during our medical universities, of course, we learned immunology, that there are T cells that can fight cancers, that there are some obstacles in fighting cancers, such as antigenic escape, for example. But here we focus something, we see something absolutely different. The way how to force antigen not specific T cells to fight cancers. This is the clinical perspective. How to force, for example, CMV specific T cell to fight with leukemia or lymphoma. That's absolutely crazy. And this is one of our current approaches. We've got also two specific antibodies, other molecules that are supposed to do the similar thing. But here, it is very useful and it's relatively easy to understand for us physicians because it uses antibody portion. We, hematologists, used rituximab for over 20 years already, hectoliters every day. So it's understandable to use something so close to us with the fragment antigen binding of very similar to rituximab, anti-CD19, CD20. Not much difference. No, it's in fact, there is a difference. And of course, another thing is the mechanism of cytotoxicity of the T cells, because to date we had experience with HLH, phagocytosis syndrome, for example, which was enormous activation of the immune system, for example, by viral infections. And here, what is absolutely new for us is the cytokine release syndrome, so the new complication of the commercially available therapy based on too high efficacy of the CAR-T cells. That's also very important aspects for us. Currently used CAR-T cells in the products, all of registered CAR-T cell products are of the second generation CAR-T cells. In fact, my presentation will be kind of wrap-up of the today's lectures. Second generation, we had so many different other molecules, but this is how the medicine behaves. Once something is produced, registered, using huge resources, it will be used till the end, till all the opportunities will be lost. Either efficacy in some indications will be not shown or confirmed, or it will move from the third-line treatment to the first-line treatment. The second step, usually in the clinic, is using this in combination with currently available products. Okay, so use the simple second-generation CAR T cells with ibrutinib, with monoclonal antibodies, with other immunotherapies, and of course this makes us more and more brave to use next-generation CAR T cells. And here is, in fact, the list of the currently registered products, both in US and in European Union. Three of them, in fact, four of them are anti-CD19 products. Tisagenlecleucel by Novartis, it's anti-CD19, and axicabtagene ciloleucel by Gilead. They are the most commonly used ones, anti-CD19, used both for lymphomas, B-cell lymphomas, and acute lymphoblastic leukemia. Then another product was registered by Gilead, Tecartus, brexucabtagene autoleucel, also anti-CD19 product, but with different name, a bit changed structure, and used just for mantle cell lymphoma, another type of lymphoma. Another company, Bristol-Myers Squibb, registered lisocabtagene maraleucel, which is pretty similar to Kymriah, Novartis, but here another approach was made to use the same ratio of CD4 to CD8 lymphocytes in the product infused. They are infused separately, CD4, CD8, but at the same amounts, and they have different products already. The recent indication, and very important indication because of the large population of patients, are products against the BCMA antigen on the multiple myeloma, very common cancer. Nowadays, the survival of patients with myeloma is about 10 years, but still many patients do not survive this time. And we've got two products registered, idecabtagene vicleucel, anti-BCMA product, and ciltacabtagene autoleucel. The names are really strange, but there are already differences because between those products in the fragment antigen binding of the CAR receptor, and one is working a little bit different than the other. So this is the current stage. But how it is when then the product is registered by EMA or FDA? It is registered based on clinical trial made in special indication, usually starting in the very late lines of treatment, like in myeloma, 4th line, 5th line, or in lymphoma and ALL is 3rd line and later lines of treatment. It means in those patients that had no chance to be cured with any other approaches. But for each indication, there must be done a separate registration clinical trial, which is, of course, very time-consuming, extremely expensive, and almost hundreds of patients need to be treated like this in order to get this registration. So currently, we've got Kymriah, tisagenlecleucel, registered for B-ALL and large B-cell lymphomas, different large B-cell lymphomas. Similarly, axicabtagene ciloleucel anti-CD19 for different large B-cell lymphomas, but look at this. Here, the line is much longer. There's already one indication in the 2nd line, registered, but only in United States, not in EMA. And there are, for example, differences indication for primary mediastinal B-cell lymphoma, and there's no such indication in tisagenlecleucel. And it means if you will treat with tisagenlecleucel primary mediastinal B-cell lymphoma, you treat out of specification products, you risk a lot, and certainly this will not be paid. Another practical approach is, of course, going into brexucabtagene autoleucel. It's a new product used for treatment of mantle cell lymphoma. Mantle cell lymphoma is another very aggressive cancer, another very aggressive lymphoma. We can treat it in the 1st line, getting very high ratio of remissions, but when it relapses, really, it's hard to treat. Therefore, we go with treatment to the 2nd line if we can. But unfortunately, it's registered first in the 3rd line of treatment. Another product, isocabtagene maraleucel, made of those mixed CD4, CD8 molecules, is also registered in similar indications. And idecabtagene and ciltacabtagene autoleucel, two anti-BCMA and anti-myeloma treatments, are registered in different way in European Union, in EMA, and in different line of treatment in FDA. Why it is like this? Usually those products first are registered by FDA, and then the companies apply to EMA for registration. So usually the indications are the same, but sometimes the experiments go faster, and it's known already, and new trials are available, that while registered at FDA on the fifth line, in EMA, it's registered earlier because the evidence was that it can work in this earlier line of treatment. So we can expect in the nearest lines that this table will be like 3 sheets of paper, because more clinical trials, more indications, so new lymphoma types. There's about 100 of different types of lymphomas. And you can move to the second line, first line treatment. There are already clinical trials in the first line treatment, and most of us believe, well, it's a good way to go. You cannot see it very exactly, but I would like to just try to sum up the efficacy and toxicity of the 3 major products, anti-CD19 CAR T-cell products, T-sargent-lecleucel, axicabtagen, and lisocabtagen. So first of all, what we would like to achieve, complete remission of the disease. So after treatment, the disease disappears, there are no signs of the disease, which gives the chance that the lymphoma will be cured. So the ratio is pretty much similar in 3 products, but a bit lower in Novartis product, Kymriah. Of course, these studies cannot be compared head-to-head, different population, a little bit different indications, but somehow we need to move with this. A bit lower ratio of complete remissions. Here you can see the rate of overall responses. So it's complete remission and partial remission, which means decrease in the cancer size of about 1/2, till 1/2. Of the cancer. So it's about 50% still in Kymriah, but much higher in Yescarta, about 90 or 85%, and in case of lisocabtagen maraleucel. So a lot of patients respond, but then they relapse, they progress, and finally we can see another issues. In what patients it works and in what patients it doesn't. Of course, all of those studies have some analysis like this, that when you have a big cancer with the big size, it will, the CAR T cells, it behaves more like a solid tumor. The CAR T cells probably don't get there enough efficiently. The ratio of tumor cells to lymphocytes is not proper. And of course, the efficacy, clinical efficacy is much lower. Another issue is that, for example, when the cancer is really clinically aggressive, CAR T cells, unfortunately, will be also a little bit less efficient than in case of less aggressive cancer. Another important point is that lost susceptibility to chemotherapy doesn't mean that we'll not respond to CAR T cells. This is very good for us, for clinicians, because we've got another mode of action of those lymphocytes. But otherwise, when we look at the population of the patients, the efficacy is more or less the same. There are no huge differences that in some population it really doesn't work and in some works in 100%, which is quite good. For example, at the beginning, it was thought that we should assess CD19 expression on the cells because, of course, it's a target. Now, we know we don't need it, because sometimes you can't see CD19 expression by flow cytometry, but even the small amounts of CD19 expressed are enough for CAR T cells to be efficient against those lymphomas. The major endpoint of such studies are progression-free survival, and for the whole population, it's this gray line, this blue, and the red line. So it's very, very similar. It means at 1 year, about 50% of patients stayed alive without relapse of the disease. A little bit lower proportion in Kymriah than in Yescarta and in Breyanzi. And of course, the ultimate endpoint is the overall survival. So still, in the Novartis trial, about 50% of patients stay alive after 1 year, about 60% in Yescarta, and the similar proportion, about 50% in Breyanzi products. But of course, the best response, a complete remission, gives the highest probability of overall survival and progression-free survival. And this is something we want to achieve. In fact, we don't want partial remissions. Of course, it gives some effect, but not leads to ultimate goal of the treatment. Importantly, diffuse large B-cell lymphoma is aggressive cancer which can be cured in 60% of patients with chemoimmunotherapy without CAR T cells. So we are fighting for the remaining 40% of patients, and we hope that these statistics will change with use of CAR T cells. Of course, another important issue when we look at the CAR T cells is the duration of the response. So how long the tumor will respond to the CAR T cells? Because at the beginning we thought the CAR T cells will stay in patients for overall during the whole lifetime, and it's not true. Unfortunately or fortunately for some patients, and in some patients the disease relapses as well. So the duration of the response in the overall population is about 60% or but higher in Kymriah than in Yescarta and Breyanzi. So it's a little bit tricky. Less complete remissions, less overall remissions, but once the patient obtains the complete remission, it is usually durable, much better than in other products. So here is already some difference. When we look at the complications, there are huge lists, and almost all patients have any side effects of the treatment. That's important. It's not very safe. Almost all patients have some features of toxicity, but it's not killing our patients like we thought before. The ratio of death related to the treatment is about 3% of patients, which is much lower than in allogenic stem cell transplantation that we do on everyday basis. Almost all of them have side effects. In the initial experiments, a relatively large proportion of patients suffered from cytokine release syndrome, so symptoms similar to the sepsis but without septic pathogens, leading frequently to lung insufficiency, to heart insufficiency, and in some patients to death. There's also another issue of neurologic complications, but the severe neurologic complications are still very rare, like in 12% or 14%. Now we compare like tisagenlecleucel with axicabtagene solaleucel, and what we see? We see more cytokine release syndrome in that product and more toxicity in that product, including encephalopathy, than in this product. And as we remember from previous slides, it was a bit less efficient here and more efficient in lymphomas here. It's always like this in hematology. The more toxicity, the more efficacy. But there is importantly difference between those products. All of them are different from each other. The first is anti-CD19 with 4-1BB co-stimulation, which according to the theory provides less activation, immediate activation, but durability of the response. In Yescarta, it's also anti-CD19, but CD28 co-stimulatory domain, which gives more acute activation but less durability. So this is why here is more immediate responses but less durability. Here is less immediate responses, but when they are usually durable. And in this product, there is low toxicity because the same ratio of CD4 to CD8 lymphocytes is given. It's of course thought that CD4 cells give more cytokine release syndrome because they are this is their biology. When we look at the other indication, clinical multiple myeloma, it's very common cancer in hematology, but what's important, there is many drugs for multiple myeloma currently. So we use first-line treatment, second-line treatment, fourth. A lot of drugs refunded in Poland. So the next drug, very dangerous drug like CAR T cells, as we thought before, was studied in the last lines of treatment, fourth, fifth, sixth, seventh line of treatment. And what can we see looking at the complete remission rate after treatment? It's 45% in the captagen vicleucel and much higher rate in another product. Both are anti-BCMA. So not the specificity may be so important, but some other features of the products. Of course, we can again look at the overall response ratio, which is very high, 80%. Of the patients will respond. Almost all patients will respond to the anti-BCMA CARs, but in case of myeloma, it means, unfortunately, most of the patients will finally relapse after this treatment. The biology of the disease is different, but still, complete remission is also something not so good as stringent complete remission, which means you cannot see myeloma with any available methods used in the clinics nowadays. So this, in theory, could mean a cure, but so far there is no claimed cure in multiple myeloma. It's a cancer which is uncurable, but perhaps CAR T cells will change this theory. And here is the progression-free survival in patients receiving the highest dose of cells. It's another thing. Both in the DLBCL or acute lymphoblastic leukemia, the response was not dependent on the cell dose. At least it was done in the very early phase trials that they declared certain range of doses and it was given. Here in myeloma, it's really important to have enough CAR T cells in order to fight the cancer, but still these steps down mean that the patient relapse. And probably when there will be longer follow-up, all of them may finally relapse. But still, comparing the products, the first one, Abecma, have about 40% of the progression-free survival at 1 year, but another, a newer one, have already about 75% of the progression-free survival at 1 year. And in this third line, fourth line, fifth line, sixth line lymphoma, it's a very, very good result. When we look at the complications, they are very common in myeloma. Very common cytokine release syndrome. These are usually elderly people with after many treatments, so it's really tricky to treat them with very high rate of neurological toxicities, but in fact no very severe neurologic toxicities and less neurologic toxicity in case of another product. So what's the difference between those products? The first one is standard second generation CAR T cell with 4-1BB domain with the standard CAR T cell receptor. But another, the newer one with the higher response to that CAR has modified fragment antigen binding based on single chain antibodies from llama targeting two different epitopes of the BCMA antigen. So one targeting one epitope, another epitope, and still and for 1BB costimulatory domain. So we can see already how it affected clinics. Much higher efficacy in multiple myeloma and less toxicity, which is something we would like to obtain. One of the standard treatments in diffuse large B-cell lymphoma to date, when we didn't have CAR T cells, was for relapsed refractory diffuse large B-cell lymphoma was autologous stem cell transplantation. So autologous stem cell transplantation is based on administration of very high dose chemotherapy, this kind of doses that will kill all the bone marrow cells, and we can introduce the healthy bone marrow taken before collected before chemotherapy to recover hematopoiesis. And recent paper showed that it may be like this if we could have CAR T cells available for each patient relapsed or refractory after initial chemoimmunotherapy, the efficacy and event-free survival would be much higher than in case of traditionally treated group of people. So these people, when they are diagnosed relapse refractory, they receive second-line chemotherapy in order to decrease the mass of the cancer, to at least 50%. When it works, then the stem cells are collected and chemotherapy given, and there is autologous stem cell transplantation. But it means that many patients will never receive this partial remission or complete remission and will not get transplant. Even when the patient gets transplant, it's most efficient when the patient has complete remission of the cancer. Otherwise, the likelihood that the disease will relapse after autologous transplant is very, very high. So it looks like, in fact, CAR T cells compete nowadays with autologous stem cell transplantation in myeloma. But looking at this, whether transplant or CAR T cell, another answer is both of them. For example, in multiple myeloma, as you saw with high rate of relapses after CAR T cells, the nowadays clinical trials use, for example, both autologous stem cell transplantation and CAR T cells given just after autologous stem cell transplantation in order to fight disease which has as little cells as possible to increase its efficacy. Another aspect is allogenic stem cell transplantation, which is more and more frequently connected with CAR T cells against acute myeloid leukemia. CAR T cells against acute myeloid leukemia target antigens on normal hematopoiesis, unfortunately. So when you use CAR T cells anti-CD33, for example, you will never have neutrophils, and the patient will die of sepsis for many, many months. So the new approaches use allogenic stem cell transplantation either to recover hematopoiesis after CAR T cells or some other combinations. Okay, another approach, as I said before, is a combination of the CAR T cells, currently used CAR T cells, with already available treatments like another drugs, anti-neoplastic drugs. For example, in chronic lymphocytic leukemia, it looks like CAR T cells used alone are not so much efficient for some reasons, and they are not registered so far. But it looks like they can be used together with another drug which is used for treatment of chronic lymphocytic leukemia, ibrutinib, some inhibitor of the Bruton kinase, especially when the ibrutinib is used before lymphocyte collection for CAR T cell production, and then after infusion of the CAR T cells, ibrutinib is administered. It looks like the efficacy is 83% remissions compared to 56% without ibrutinib, and toxicity is lower. It's thought that some other treatments may affect the quality of lymphocytes that are used for production of the CAR T cells. So this is another arm of studies. For example, combination of lenalidomide with CAR T cells, so in other immunomodulatory agents, or lenalidomide with anti-BCMA CAR T cells for multiple myeloma. But okay, we are still playing with the second generation, and why not third generation? Why not this fourth or next generation or CAR T cells yet? One, because of availability of the treatments. But our current treatments have a lot of problems. First, limited efficacy. It doesn't work in all the patients. It has significant toxicity. We don't like toxicity. The production process is usually long. Failures of this process happen, and still we can't use it in certain types of cancer. So these are, of course, there are many more problems which we need to face. So why not third generation CAR T cells? They have two costimulatory domains, CD28 and 41BB. So they should be more and more efficient. And it looks like how they get activated and proliferate in response to the antigen ex vivo. This is high dose of the CAR T cells, which expand so much. This is third generation CAR T cells compared with the second generation CAR T cells. Okay, great. When you give very low number of CAR T cells compared to the target antigen, only third generation CAR T cells get activated and proliferate, while there's no enough number of second generation CAR T cells. Okay, so great. We could use third generation CAR T cells when we have just few cells of the lymphoma that will be responsible for the relapse to kill it. another thing, we can use it in very small numbers so that the production process will be shorter. But unfortunately, I don't know any studies that would show that they are in fact clinically better, even in the murine models, more efficient and less toxic than second generation CAR T cells. Don't know why it is. In theory, they should be, but so far, to my knowledge, nobody generated such a study that would definitely show the third generation and two costimulatory domains are better. Probably the biological knowledge is changing, like Dr. Kolanowski said before. Too much activation sometimes gives exhaustion and less survival of the CAR T cells and many, many other problems in the clinics. So, what in the future? What we would like to use in the future in order to fight our cancers? Because there is different nomenclature about the CAR T cells, but I would like to mention just a few different kinds. Armored CAR T cells, discussed by Dr. Kolanowski, the most important for me population are those cells that are fighting the normal inhibitory signals that inhibit CAR T cell activation in the organisms. Another track CAR T cells is this kind of CAR T cell that can carry large volumes of the transfected DNA that is doing some other functions. Universal CAR T cells, so such CAR T cells that can be put on the shelf and used in every patient from the same donor, or switchable CAR T cells, those you can either switch on, turn on, or switch off when there is too much toxicity. But what is the proportion of those new next generation CAR T cells in clinical trials? The majority of clinical trials still are done with switchable CAR T cells, because at the beginning everybody was afraid of cytokine release syndrome, and every product started to have this kind of switch off mechanism. Another large proportion of the CAR T cells used in the clinical trials are currently the track CAR T cells, so carrying another transgene. Starting from armored CAR T cells. This is the real problem in the clinic when the tumor environment stops normal CAR T cells from their activation through the immune checkpoint proteins. And armored CAR T cells use this as opportunity to get activated. For example, the tumor cell wants to inhibit CAR T cell through the PD-L1 ligand. But here, when the CAR T cell is modified with activation domain instead of inhibitory domain connected to the PD-1 receptor, then the more cancer tries to inhibit the CAR T cell, the more activation the CAR T cell gets. Another approach is modification of the CAR T cells with nanobodies or fragment of the antibody Fc connected with the PD-1 receptor. They are secreted and they just inhibit PD-L1 at the cancer site. Another, of course, opportunity is to downregulate expression of PD-1 in CAR T cells using CRISPR mechanisms. And those CAR T cells are already used in clinical trials. Another track CAR T cells are those that are modified, for example, with genes carrying cytokines like interleukin 12, 7, 15. Why they should secrete cytokines? For autocrine activation and prolonged survival of the CAR T cells, but also in order to stimulate immune system of the patient, either specific immune system and non-specific immune system, because not only CAR T cells fight with cancer, still there are specific T cells, NK cells, macrophages that can also do their job against the tumor. Switchable CAR T cells. Another portion of the trials with CAR T cells are these switchable CAR T cells. The major goal of switching off is to limit toxicity. Once you get too much activation, too much toxicity, you can just turn it off. One way how to do it is, of course, by modification with some surface antigen like EGF receptor. Then addition of the monoclonal antibody to the patient can kill CAR T cells and you get rid of toxicity. But the most common strategy was modification with inducible caspase-9 transgene, so that administration of the drug switches off the CAR T cell when there is too much cytokine release syndrome. Another approach is modification with herpes simplex thymidine kinase, which can be turned on by administration of ganciclovir in patient, and then CAR T cells die. Importantly, there's only one case of using this mechanism described in the literature. Usually, we don't need it. It appeared that we don't need this switch, this negative switch. We can pretty well manage the toxicities in majority of patients, and so far the new products usually develop without this negative switch. Another portion of the CAR T cell products are universal CAR T cells. So currently we use product produced for certain patient. But in the future, we would like to have universal product that is stored in our pharmacy. And when we have patient, we just apply for the product to the pharmacy. We don't need to collect T cells, wait for 3 weeks, and then for transportation of the CAR T cell, because the clinical condition of the patient may change, that he may get infection, will never get CAR T cell treatment. So this kind of universal CAR T cells would be possible by knocking down the TCR receptor, so they will not cause graft versus host disease, and knocking down MHC molecules, so they will not be eliminated by the patient immune system. Of course, this happens by a genetic modification or other strategies, as discussed before. This strategy is also important when you want to produce CAR T cells against the T cells, because you have T-cell lymphomas and they have T-cell antigens which are common with the CAR T-cell surface. So one of the strategy is decrease or expression of the certain molecules on the surface of CAR T-cell, the molecule that is also the target on the target lymphoma surface. And then it's possible to use anti-CD4, anti-CD9 CAR T-cells because they don't kill each other. Other strategies of CAR T-cell therapy, CAR and K cells are very intensively investigated. There is one very good paper, but in small numbers of patients, showing that they are very efficient. They have many limitations, such as a limited expansion possibility of those cells. But there are also many opportunities. You can use them as universal products, because NK cells are more prone to or resistant to these allogenic responses, for example. They have also different killing mechanisms, and there are many enthusiasts of NK cell CAR T cells. Still, the majority of the trials are on CAR T lymphocytes. Another intensively investigated approach in clinical trial are bispecific or dual CAR T cells. So bispecific have chimeric antigen receptor with two different antigen binding sites, like in this multiple myeloma second product, two different epitopes or two different antigens. And dual CAR, they have just two different receptors, two different CAR T cell receptor against different antigen. Why two different antigens? First, to avoid antigen escape. Another is to target different population of the cells. For example, the mature lymphoma or leukemia cells and stem cells of the lymphoma and leukemia. So this is already very commonly used in the clinic, and there are no obstacles to use it. Based on those modifications, for me, naive physician, it would be great to have such a universal CAR T cell on the shelf, so I would not need to collect the cells from the patient. This will be armored in some cytokines or perhaps chemokine receptors that will show the way of the CAR T cell to go directly to the cancer. Finally, it will have some substances some other opportunities to stop immune inhibition, and also another thing, to have something to turn off when you don't need it. But there are already so many modifications, as previously said. Probably this is not the way. So many modifications in single cell. Probably the solutions are more simple, because as we see in the clinics, the way to develop CAR T cells was very long. But what happened? at one time some new development comes, new idea, new technology, everything changes. And this is also probably the same for CAR T cells against the solid cancers. We can try and think and think and think, but one day something will be found that will break this barrier. And so, in fact, we don't know what will be the future of the CAR T cell treatment. We know what is the presence that we use CAR T cells in many clinics. We started use it also in September this year. We're getting more and more experience. We are more and more brave to using this kind of complicated CAR T cell therapies. Another important issue and question is whether they will be produced locally, like we do for bone marrow transplantation right now, because at the beginning everybody thought, yes, we will produce our CAR T cells at our facility, but it's not so easy, also according to the regulations. Or this will still be drug, because it's drug once it is registered and produced by independent companies. Currently, to my knowledge, different academic programs stop at this either preclinical trials or phase 1 clinical trials. As said before, they cannot develop larger studies that lead to registration. And currently, in the current world, you can't use this product without registration. There is another option, clinical trial, and hospital exemption, when the principal investigator takes all the responsibility that everything that happens with treatment with this product will be his fault. So we cannot just produce it for us right now, and so probably this will be the future. One of the stopping mechanisms for today is the price. Very high price, because the technology is complicated, the procedures are very complicated, and it's very unique orphan application. But the price will surely go down like it does with many other drugs. And so that those products will no longer be orphan products, will be used more commonly, especially when they will start to use it in autoimmune diseases, so in millions of people, or many other indications, like many other immune indications. Then it may become a treatment not only for hematologists or oncologists, but for like every physician. Thank you very much for your attention.
For this excellent overview of the current opportunities and challenges of the CAR T therapy in the clinics. So I would like to open the discussion. So perhaps I will start. At the beginning you mentioned this time course of introducing CAR T in the therapy registration of new drugs. So it's for the sake of safety and ethical point of view, it's of course obvious that you start with the last chance patients. But once you see that it actually works, yeah, and that it could be an option in early lines of treatment, so is it then still ethical not to give the opportunity to the patients, and are there any shortcuts in these procedures? How does it work?
Of course, there's always a balance between the efficacy and toxicity, which we need to consider, and safety for the patient, which is the most important. Sometimes we've got very efficient treatments which are already established, and so far there is no clear opportunity for the patient to be treated with CAR T cells with having like larger risk, because it's still unknown. Everything needs to be confirmed in clinical trials, but it goes very fast to the first lines of treatments, because there are two strategies: to leave the drugs for the next line treatments so that you can prolong the survival of the patient every 2 years, every 3 years, every 6 months, and you give another drug more and more potent. But another strategy to use everything you have at the beginning as strong as possible in order to obtain as deep remission as possible, so that the patient will survive 10 years, 20 years without treatment. Maybe will lapse after that time, but maybe not. Currently, we can't use it because it's not registered in the first line trials, but in first line treatment. But when there is clinical trial, depending on the protocol, but I would not hesitate much.
Thank you for this great talk, Professor Basak. You discussed about the strategy, how to produce the CAR Ts. Should it be in the clinics or should it be rather like in independent companies? My question would be for a strategy for development. Do you think that CAR Ts, because right now they are used only in the big clinical centers, do you think that this will be spreading in a way that CAR Ts would be like available in every hospital, that has a hematology department, or you think this rather would be like clinical centers that keeping this technology and that like patients are treated in there?
That's an excellent question. Being the chair of the department, I should say no, only in specialized centers and in my center, not in the other center. But in fact, at the beginning, everybody was afraid of this treatment. Strange complications, intensive care, very close to the department. But finally, we saw that we do much more aggressive treatments than CAR T cells in our clinics. In fact, I don't see many theoretical problems with treating with CAR T cells in every hospital. Of course, important thing is taking care of the finances of those hospitals and the procedures. So sometimes in some centers the procedures work better, in some not. In some there are no procedures, and still part of this process needs to be strictly regulated so that even the product will be this product and not the other product produced based on the patient cells. Another is, of course, keeping an eye on the finances, of course, related to these treatments. This is probably the most limiting factor to spread across different hospitals. But yeah, I think not only hematologists will use it in the future.
I would like to ask you about your opinion. How far are we from introducing T-cell therapy, CAR T-cell therapies in the treatment of T-cell malignancies? Mm-hmm.
Many preclinical trials, very efficient models, and there are also clinical trials. To my knowledge, I think maybe somebody has some more data. None of the results are so optimistic so that it will lead to immediate registration of those products. Still, I saw so many probable ways how to solve the issue of the fratricide between the CAR T cells, but somehow none of the strategy is leading nowadays, to my opinion. I don't know, perhaps somebody knows, maybe Tomek knows something more. Yeah.
Quite tricky. Maybe different cell type that we can, from my side, maybe different cell type that could be used in this particular way, because it's anti-CD3, basically. That's the easiest way. But on the other hand, you need to be more specific, because you cannot get rid of all CD3. Yeah, so it's a complicated story, much more than CD19.
Depending on the antigen, perhaps CAR and K cells for T-cell lymphomas would be better.
So if there is no more question, and I have one comment. Okay. So it was really nice to see that our molecular engineering has really an impact on the efficiency in patients. And I have one more naive question. Do you think reasonable to collect the patient peripheral blood before the treatments, the regular treatment starts, to avoid this manufacturing failure?
Yes, we do it currently, but perhaps not before the first-line treatment, but already in lymphomas, after failure of the first-line treatment in younger patients, we collect peripheral blood T cells in order to produce CAR T cells made of fresh, not exhausted T cells. So it became our practice, but at the diagnosis, sometimes there's no time. When it's an acute condition, there is no time to collect, to do the apheresis. Sometimes there is leukemia, and what you collect will be just the leukemic cells with very few T cells, but sometimes it's also done like this. Maybe one day every patient will have some sample collected for different purposes, like biobanking at the beginning. Now we need the whole apheresis with the lots of T cells, but with the nowadays techniques, you can produce like smaller amounts of CAR T cells that will expand in the body already. So you don't need apheresis probably in the future. Maybe in some patients, for sure.
Thank you. Are there any more questions? If not, we would like to thank you
very
a lot
much.
for your lecture. And here's a little gift. And I will give a word to oh.
Okay, so I think the time has come to close and wrap up today's event. It was already the second Hematology Day organized within the Next Level project, which is financed by the European Union Horizon 2020. And thanks to our excellent speakers, we had an opportunity today to hear about the CAR T therapy from different perspectives. So in layman words for the students, about the most important and urgent issues from the patient's perspective, and also about the clinical and scientific challenges that need to be addressed in this field. So let's thank our speakers once again, Professor Lidia Gil, Professor Grzegorz Basak, Dr. Tomasz Kolonowski, and Magister Marta Kasprzyk. And we would also like to thank you all for taking part in this event. It was a great pleasure to host you here, both people here on site and online. Thank you for your participation. And perhaps it is too early to say that, but maybe not, because it already emerged in the corridor discussions, that I have a feeling and hope that this event may be continued beyond the duration of the Next Level project. Well, we will see, but I think it's a good starting point. So at this point, I would also like to cordially invite you to the next year's Hematology Day, which will take place next year, and will be something about Hodgkin lymphoma, I guess. So thank you once again for this whole day today.